ones that are unchanged.
Robert.
After you write down that word, I want you to look at the board and see where I point off the parental chromatids. Notice, please, there is a chromatid going along here that undergoes no crossover. The crossover is between this chromatid and this chromatid. So that one remains a parental. All right? And then notice this one here doesn't participate in any crossover, and so it remains with the parental sequence. So this one is A, B, C, D, E, F, G. And this one is the inversion A, B, E, D, C, F, G.
So Dr. Shepard, when we put our cross or X for the crossing over, we're always going to be doing between just the homolog, right? Not.
We're always doing crossover between homologs, not between centromeres, or not between sister chromatids, because a crossover between sister chromatids is genetically silent. There's no sense doing it. Now comes the fun part. If you've got colored pencils, this may be the time to pull them out. Watch what happens here. We start here,
Kim, this is gonna be money
Go up
in a minute
there.
after.
down here.
Oh, excuse you.
Notice what we've got now. A, B, C, D, E, B, A. And now it's worthwhile putting in the centromeres. Here's one, here's one, here's one, and here's one. Notice what we've got here. We've got a chromosome that is dicentric. It has two centromeres.
Is the dicentric the one that crossed over?
Right here, yes.
And it's not a parental, right?
Well, it's still paracentric because the centromeres are outside of the inversion sequence.
Yeah.
Kim,
So far, so good? Now let's go, because Christmas is getting near, we'll do red and green. All right?
Oh, well, he's just saying that that's the original sequence.
Now we start over here.
So anything, yeah.
Watch what I'm doing here as I do this.
Theoretically.
That one has no century.
Notice,
really crazy.
where are the
smart.
centromeres? Ain't any. So it's called acentric.
What? I don't give a shit.
Yeah. Let's
That
see.
phone
Let's
is
go
in his
here.
head.
Let's start here. Yeah, I started there. G, F, E, D, C. You can do it either way. You can go in either direction on this. I always tend to start on the second one on each side, just as a convenience. You can always start down here. Whichever you choose to do, be consistent on these two recombinants here. If you do one starting on the second, do the other one starting on the second. If you do the one on the fourth, do the other on the fourth. Okay, now you've got two centromeres. Is that viable? Basically, you're gonna rip the thing apart when you go into anaphase. All right? Is this viable?
Can't pull it apart.
You can't pull it anywhere. It's gonna be lost. It just goes randomly one place or another. Has no centromere. May not even end up in the nucleus. It could be in the cytoplasm and be degraded. So, both of these
Tripod.
Dude, does it have a stand on the thing? Is it curious?
are non-viable.
Because 2 centromeres and no centromere?
Yeah. Notice what your textbook shows here. They show the anaphase right here in the middle of the figure there. And if you want to learn it in that form, you can. To my eye, that is messy and complicated and a pain in the butt. I'd rather just go and list the 4 resulting chromatids and ignore the fact that they are attached to each other.
How come those last two are non-viable? Because it's acidic and...
Okay. Can this thing be pulled into both daughter cells?
No, because it has 2 centromeres.
Yeah, it's got 2 daughter cells. It's going to attempt to do exactly that.
Yeah.
That's what they're showing right here. They're being pulled apart. You end up just snapping the thing in 2. This one's not viable because it never ends up in the nucleus. It never gets pulled out over to the 2 new daughter nuclei.
Okay.
Either way, it's a non-starter. At this point, the cell is dead. Yeah.